Identifying the 5 types of leukocytes, that is, white blood cells, can be fun, but at times frustrating. When scrolling across the histology slide, you mostly see red blood cells that are vastly outnumbering the white blood cells, making it difficult to find and distinguish the white blood cells. Well, I have one very simple rule for you. Never let monkeys eat bananas. Why is that, and what does that have to do with blood? Find out what it means in tips and tricks in this video on how to identify white blood cells. If you want to skip ahead to the practice questions, the questions are timestamped below in the video description. But for those of you wanting a bit more of a mini lecture on how to distinguish the white blood cells, or simply want to know why you should never let monkeys eat bananas, simply put, it is a mnemonic and helpful way for remembering the relative number of different types of white blood cells, also known as leukocytes. Leuko referring to white, cyte referring to cell. The order of relative quantity, that is how likely you are to encounter them, is as follows. Never. Neutrophil. The most common white blood cell you'll encounter at 40 to 60% of all white blood cells. They are very distinct and easy to identify, having a multilobulated nucleus. Lymphocyte. The second most common white blood cell you'll encounter at 20 to 40% of all white blood cells. They have a relatively large nucleus with a very small portion of the cytoplasm visible. Monocyte, present at about 2 to 8%. They are a bit uncommon but easy to distinguish because they are the largest among the white blood cells. Eosinophil, present at roughly 1 to 4% of all leukocytes. They're rather uncommon but very easy to distinguish due to their reddish appearance. Basophils. These are rare and relatively hard to distinguish between lymphocytes due to their dark staining appearance. So these are the 5 types of leukocytes in the order of how common they are. Depending on the source, the relative percentages might be a little bit different, but they can be fairly subdivided based on their appearance, such as being granulocytes or being agranulocytes. So all granulocytes contain small granules of proteins and enzymes that stain to help aid us and distinguish what kind of leukocyte they are. So here we see these little dots in each one of these images. Those are the granules. With neutrophils, they get their name because the granules have a very neutral stain to them, where they only stain slightly purple, very light. Those granules can be very hard to distinguish. The key thing about neutrophils is their nucleus here is multi-lobed. So we see 1, 2, 3, and 4 lobes in this one. It can vary from 2 to 6, with 3 lobes being the most common, but we can also see some with uncommonly 5 and rarely 6 lobes. Unfortunately, immature band neutrophils can be observed in peripheral blood, where instead of having these lobes right here, it's more horseshoe shaped and not quite as lobed. Eosinophils. So the granules here stain best with a commonly used acidic eosin stain, thus they get their name eosin for the acidic part and phil for loving. Because of that color, they are pretty easy to identify. Their nucleus is most often bilobed right here, so there's 2 of them. So it appears that there are 2 nuclei, but they're really connected right here in the middle. Overall, they kind of have this alien head-looking appearance with 2 large eyes and a large head. Basophils. As far as their name goes, their granules right here, they stain best with basic or alkaline stains, thus baso for basic and phil for loving. These are going to be the hardest to distinguish and identify. First, because they're the rarest of the 5 white blood cell types. Second is because their granules stain dark and they stain very close to the nuclei in color. So their nucleus is often more bilobed or S-shaped. But the problem here is because of all these granules are very similar in color, they overlap and it's really kind of hard to see which is which. But we'll go over how to really identify it in this video. Lastly, note that all 3 of these cells are just a bit bigger than a typical red blood cell, and if you were to measure the diameter of the cells, and we'll go over that too as well with some examples. The name agranulocyte is a bit of a misnomer. A meaning without or none is not entirely true. They still have granules in them, but they're a bit smaller and don't stain well, so we can't always see them. However, it is a convenient way to categorize these 2 cells. So our monocytes are going to be the largest for white blood cells, and they're about 2+ times the diameter of a red blood cell. So if you look at the diameter here compared to here of a red blood cell, if you think about area-wise, you could look at the total area and say, hey, I could probably fit about 4 or 5 of these red blood cells inside of this single monocyte. Monocytes tend to have this horseshoe-shaped nucleus, but do note that neutrophils, if they're undeveloped or underdeveloped, they can kind of have this shape as well. So we have to really rely on size oftentimes. Then our lymphocytes over here, they are our second most common white blood cells, and they generally are slightly larger than red blood cells, but they can vary in size and some can be quite large. One thing to note though is the diameter, overall the area of the nucleus here in a lymphocyte, is roughly the same as the total area or diameter of a red blood cell. So they are also the only formed element, that is red blood cell, white blood cell, or platelet, that has a different embryological origin, but they still play a vital role, as do the others. Also note that both of these cells, they have a single or unlobed nucleus. So that's really key here. So having gone over all the basics of our 5 cells, let's take a look at each cell at 400 times power magnification. So here we see 7 leukocytes that are surrounded by many more red blood cells. The red blood cells have lost their nuclei, so they appear donut hole shaped in the middle, and therefore they don't stain purple. These smaller purple dots, those are our platelets. Now, all 7 of these are the same leukocyte. In all cases, they're a little bit larger than a red blood cells that surround them, and they have multiple lobes. This one has 4, here's another one with 4, and so on and so forth. Given that and the fact that there are so many of these and they're all the same type, remember our never and never let monkeys eat bananas, we have neutrophils. Note that they are granulocytes, but they can be difficult to see, especially at this magnification. This neutrophil over here appears to be a bit more immature, and that is a nucleus isn't fully lobulated into 2, and it can be mistaken for a monocyte. I've never asked one of these on a lab question, on an exam, because this can be a bit confusing and a bit of a gray area. But do know that these immature band neutrophils, they can be observed a bit in peripheral blood, so be careful with those. Now this image has been cropped and zoomed in a little bit. First thing that stands out is there's a single nucleus in this leukocyte, and that single nucleus is roughly the same size as any one of these red blood cells that surround it, with the entire cell just being slightly larger than a red blood cell. So right here is the cytoplasm, and overall it's not granular because we can see it, there's no dots that are visible. And with that should let us know that this is a lymphocyte. It is our second most common cell, than the let in never let monkeys eat bananas, a lymphocyte. First thing that should stand out about this leukocyte is it's relatively large compared to the red blood cells that surround it. Diameter-wise, there's at least 2 or more times the diameter of this leukocyte compared to our red blood cells, or area-wise, we could fit about 4 or perhaps 5 of our red blood cells inside of our leukocyte. Then there's a single non-lobulated nucleus that's horseshoe-shaped, therefore we have a monocyte, the monkey in our mnemonic. Note that they can appear a bit granular depending on the sample, but this is an agranulocyte. The next 2 are granulocytes, and the granules are a bit more apparent. The color of this one should stand out immediately to you. The granules here stain more acidic, so with that alone you should recognize this one as an eosinophil because of its bright pink appearance. Now it does have a bilobe nucleus with a small little bridge right here that connects the 2 lobes together, and it kind of looks like an alien face. This is our EAT, or the second least common leukocyte from our mnemonic in never let monkeys eat bananas. There are actually 2 different types of leukocytes here. The bananas in our mnemonic, that is basophil, is our rarest leukocyte and is the one over here on the left. I have a lymphocyte here on the right for comparison purposes because they can be easy to confuse. The lymphocyte has a single round nucleus, and that nucleus is about the same size as a red blood cell. But we can also see that there's a clear ring on this side as well as the other side that can be described as a ring or a halo. We see that because there are no granules, the stain of the basic dark purple nucleus here is easy to distinguish, and the cytoplasm, because there are no granules, it appears clear. With our basophil here on the left, the nucleus is not easy to distinguish because there are numerous basic staining granules that are superimposed over that nucleus. Now, if this set of examples was a bit difficult due to clarity and magnification, the next set will use oil immersion at 1000 power, and I have a little bit of a hack on how to identify the rarer leukocytes for you. Here's a sample of blood taken from the Buffy coat. Now, what exactly is that Buffy coat? So if we were to take a sample of blood and centrifuge it, that is, spin it at a very high speed, the contents would separate based on density. At the top here we have plasma, which is mainly water density. At the bottom we have heavy hematocrit, which is red blood cells. They're filled with iron. And right here in the middle we have the Buffy coat, which is mainly white blood cells and platelets. But well, because it's very thin, we're also going to get a bunch of red blood cells in there, just based on how we get the sample. So this is going to make finding monocytes, eosinophils, and basophils much easier to spot since the relative amount of leukocytes is much, much higher. From here on, in the rest of the video, has practice questions. I'll show each slide for about 10 seconds, and then I'll post the answers and give a quick explanation as to why they are that way. If you need more time before the answers appear, just pause the video. In each sample, there will be multiple types of leukocytes. Here we go. So all of these purple dots, those are our platelets all over the place. These red circular shaped donuts, those are our red blood cells. And these first 5 leukocytes here on the left are all lymphocytes. Now, why is that? If you look at the nucleus on each one of these, the nucleus is about the same in diameter as a red blood cell, and we can see a little bit of that cytoplasm on the outer parts, although they each vary to a degree. Our last one over here is a neutrophil. We can see that there are multiple lobules for the nucleus, and the cytoplasm, it is granulated, although that isn't a very good indicator. So we have a neutrophil here. So the first thing that should stand out in this cell on the left is overall it's a relatively large cell. We could fit a bunch of these red blood cells inside of this single cell. The next thing should stand out is this large nucleus that's U-shaped or horseshoe-shaped. So overall, this cell right here is a monocyte. Compared to our cell on the right, it also is relatively large, although they can be a little bit smaller than a monocyte. Key thing here, though, is we see 1, 2, 3, and 4 lobules in this nucleus. Therefore, we have a neutrophil. So here we have a lot of acidic staining granules. They stain pink. And we have a bilobe nucleus connected by a bridge here in the middle that's not easy to see. Overall, looks like an alien head. Here we have an eosinophil. Compared to the cell over here on the right, the granules are there, but they don't stain quite as well. They're more neutral staining. Here we see a few different lobes in that nucleus, although they're superimposed and hard to see. Here we have a neutrophil. The cell here on the left compared to the other two on the right, it's relatively large, so therefore you can fit a bunch of red blood cells inside of it. The nucleus right here, there's a single nucleus. It's somewhat horseshoe shaped, although it's kind of hard to distinguish. This right here is our monocyte. Over here we have a 3-lobed nucleus, 1, 2, 3. We can see a little bit of cytoplasm there, a bit granulated, hard to tell. We have a neutrophil. Here we have a nucleus, and that nucleus is about the same size as a red blood cell, and we see a little bit of that cytoplasm, more like a ring or a halo. Here we have a lymphocyte. Here we have acidic staining granules, as well as a bilobe nucleus connected right here in the middle, kind of looks like an alien head, we have an eosinophil. Over here, we have a single nucleus, and it's about the same size as a red blood cell, although it looks a little bit larger compared to those. This one might be a better representation. We see a little bit of the cytoplasm on the outer part, like a ring or a halo. We have a lymphocyte. This white blood cell is a little bit larger than the two on the right. It has a little bit of a granulated cytoplasm, but we have a nucleus that is lobulated, 1, 2, and 3 lobes. Overall, we have a neutrophil. Now, the next two might appear very similar, and they are. If you see this one here, the nucleus goes from there to there, and we can see a little bit of cytoplasm over here. Compared to the one over here on the right, there's not much of a cytoplasm we can see. You can see there's granules all over that, but you can really see more of a distinct nucleus here on the one on the left compared to the one on the right. That's because the one on the left is a lymphocyte. We can see the cytoplasm here. And the basophil over here on the right, that nucleus, it's really hard to distinguish. It's kind of this S-shaped thing, but we can't see it because there are a lot of granules that are superimposed over that. So that is our basophil. Here we have one of the smaller white blood cells, although it can be a little bit larger. We have a single nucleus. It could be about the same size or bigger than a red blood cell. A little bit of cytoplasm here visible, like a ring or a halo. We have a lymphocyte. Here we have acidic staining granules as well as a bilobed nucleus. Overall, kind of looks like an alien head or face. We have an eosinophil. Next, these two are the same cells. We can see that it's a bit larger than our other white blood cells over here, particularly this one. We can see there's a little bit of granules that stain neutral in the cytoplasm, and looks like there's about 2 lobes to each one of these. They can vary in shape and size. Here we have 2 neutrophils. The bottom 2 leukocytes are the same cell types. Overall, note that there's a single nucleus in both cases, and overall they have this U or horseshoe-shaped nucleus. The one on the right has a little bit of indentation right there. It's kind of hard to see. Overall, the size of these cells are a bit large, and they are kind of irregular, which is typical of monocytes. The one up here, again, notice the nucleus, single. Overall, it's about the same size as a red blood cell. We can see a little bit of the cytoplasm right there, ring or halo shape. We have a lymphocyte. Here on the left, we have a leukocyte. You can kind of see a granular appearance from the cytoplasm, but the big key thing here is lobules, 1, 2, 3, 4, and possibly 5. It's superimposed. With that, we have a multi-lobulated nucleus, neutrophil. Over here, again, overall diameter equivalent from the nucleus to the red blood cell, a little bit of a cytoplasm, we have a lymphocyte. So with this leukocyte over here, if you squint your eyes and look in the middle right here, perhaps you can see overall the outline of the nucleus. It doesn't quite look S-shaped or bilobed in this case, but we can see there's these areas of darker staining granules that stain dark purple or basic. Therefore, we have a basophil. Compare that to this leukocyte over here that's pretty easy to confuse the two between. We can see overall the nucleus, but we can see the cytoplasm here to the side where we don't see those granules stain quite as apparent. Therefore, we have a lymphocyte. Now, these two are very commonly mistaken in lab, but hopefully this video has given you some hints on how to distinguish the two. Now, just remember though, if we follow our mnemonic, the never let monkeys eat bananas, that's going to help you to identify these by knowing the relative percentages of them. With neutrophils, the never, they're all over the place. Lymphocytes are let. We're going to see those as well quite a bit. Right in the middle, we have monocytes, or monkeys. Those are a bit larger, easy to distinguish. Even easier is our eat, or our eosinophils. Those are very easy to distinguish because of those acidic looking granules. And then lastly, we have our basophils, which are the most rare of them all. Those are our bananas.